Hello, it's me again. Alright, so let's jump right back in. So in these parts of the lesson, we are going to learn about osmosis. Right, the learning outcomes: define osmosis, investigate and describe the effects of osmosis on plant and animal tissues. Discuss the factors that affect the rate of osmosis. Alright, so the cell membrane is a partially permeable membrane. It allows some substances to pass through, but not other. The figure shows two arms of a U-tube, separated by a partially permeable membrane. This membrane is permeable to water molecules, but not to sucrose molecules. Sucrose is known as table sugar. So here we have an area with 10% sucrose solution. Right, these are the sucrose molecules. That means that out of maybe 100 ml, 10% of it is actually sucrose. And here we have 5% sucrose solution. There are fewer sucrose molecules here. So logically speaking, what is the percentage of water in A? We take 100% minus 10, which means that this area is 90% water. And here is 95% water. Now, because the sucrose molecules are not able to pass through the partially permeable membrane, the sucrose molecules will stay in their own region. Those in A will stay in A, those in B will stay in B. However, the water molecules can pass through the partially permeable membrane. So what we see is this. There will be a movement of water molecules from B towards A. Okay, to be more specific, there's actually a net movement of water molecules, which means that there's an overall movement from B to A. So why is that the case? Remember the math that I just explained to you? Here is 95% water, here is 90% water. So just like what we learned in diffusion, there's always going to be a movement of molecules from a region of higher concentration to a region of lower concentration. And it's the same for water. Okay, so for water, it's very specific. We can only use osmosis for water and nothing else. So water molecules will move through the membrane. Okay, so what has happened? Right, sucrose molecules cannot diffuse from arm A to B because the molecules are too big to pass through the pores of the membrane. Water molecules move from B to A because solution B has more water molecules per unit volume than solution A. A dilute solution has more water molecules per unit volume than a concentrated solution of the same volume. The net result, both arms A and B have the same solute concentration, right? The number of solid molecules per unit volume. Okay, so technically, osmosis is a certain form of diffusion. Okay, but just to avoid being confused by that, let's just try and keep them separate in our minds. We just say that whatever happens to water molecules is osmosis and everything else is diffusion. Okay, when I say water molecules, I mean liquid water only. Alright, for gaseous water, actually it is more accurate to say that for water vapor there will be diffusion, and for ice it's unlikely to have diffusion or osmosis also, unless it melts. Okay, so this is mainly for liquid water. Okay, so here we have a simple experiment to show osmosis, right? And this is something that we can do in the lab too. A boiling tube is basically a bigger version of the test tube, and you have distilled water and you have sucrose solution inside a dialysis tubing. Okay, another word for dialysis tubing is whisking tubing. Please write this down. V-I-S-K-I-N-G. Whisking tubing. Now this whisking tubing / dialysis tube, right, is a partially permeable membrane. It's made in a factory, so it can have different settings, right, different sizes, whether you want to be very, very small holes or very, very big holes. But most of the time, the size of the pores are usually similar, right, to a cell membrane. It's designed to be similar to a cell membrane. Okay, so let's see how to do this experiment. Okay, you can read the rest yourself. Basically, after 20 minutes, right, what will we observe to the level of liquid inside and outside the tubing in the experiment? So what happened to the sucrose solution, the volume, and what happened to the distilled water, the volume over here, these two lines, the darker blue and the lighter blue line. What we should see is that there will be a net movement of water molecules into the reskin tubing, and that will result in the increase of volume in the center line upwards and a decrease in volume for the water line over here. So it might drop a little bit and this part might increase a little bit. Okay, so yeah, so this is the experiment that can show osmosis. It's a bit finicky, so sometimes it's hard to set it up exactly how you want it to get, and it may or may not always work. It should work, right? But depending on how you tie the knots and all that, sometimes it fails. Okay, so when we do this, we gotta be very careful. Okay, so let's learn about this word called water potential. Now, the term water potential is always used in connection with osmosis. So you cannot use the word concentration for osmosis, right? It's very specific. You must use the word water potential. Why? It's just science, okay? Scientists want it to be this way. So as students, we have to do it like that, all right? So what is water potential? It's a measure of the tendency of water molecules to move from one place to another. A dilute solution will have a higher water potential than a concentrated solution. That means that the water molecules in a dilute solution are more likely to move than the water molecules in a concentrated solution. And so, water molecules will move out of the dilute solution and into the concentrated solution through osmosis. When a partially permeable membrane separates two solutions of different water potential, a water potential gradient, okay, so it's slightly different from that concentration gradient, right? It's called water potential gradient. It's similar concept, but the words are different. Okay, so this water potential gradient is established. Water molecules will move from a region with higher water potential to a region with a lower one, down the water potential gradient. Okay, so here we see this area is more solute. That means, what's a solute? A solute means something that can dissolve in water. For example, salts, for example, sugars, right? So previously they used sucrose. Another common solute would be salt, table salt, NaCl. Then over here, this is less solute and high water potential. Now, of course, the thing that has the highest water potential, of course, will be pure water, right? Distilled water or pure water, the same meaning. That basically has no solutes inside, so it's 100% water. That will be the the liquid that is highest in water potential. So here, what we see is that the water molecules will move down the water potential gradient through this partially permeable membrane. The thing about osmosis is that it must have a partially permeable membrane. If you do not have a partial membrane, then osmosis cannot take place. The liquids will just mix normally, right? Just like how you imagine the liquids can mix when you pour two liquids together. That's exactly how it happens. But when you have a partially permeable membrane, then you prevent the mixing of the liquids and only allow the water to pass through. So please take your highlighter and your star, put a star here, and this is the definition of osmosis. Memorize. Test yourself on this. Osmosis is the net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane. Okay, every single word here is important, even the word net. Okay, so we've seen this picture before. Let's look at this thing. Okay, this is called the new water plant. They use this thing called reverse osmosis. It's not exactly the same as osmosis. There's this they add pressure and everything to it. So I'll skip it. Okay. Let me just silence my phone. Alright, so osmosis can be affected by three factors: the water potential gradient, the distance over which water molecules need to move, and the surface area to volume ratio. Now, okay, when I read this word distance, right, I realized that I missed out one thing in the previous in the previous lesson on diffusion. So let me just sorry, I keep messing up this because my slides are, the page is all over the place. So I just want to quickly recap a bit about diffusion distance before we can continue with osmosis. Okay, so what is diffusion distance? Diffusion distance is basically the time taken for a substance to move between one point to another point. Okay, the shorter the diffusion distance, the less time is needed for the substance to travel, and so the rate of diffusion is higher. Okay, so let's look at some examples right over here. Right, so this is in the lungs. In the lungs, there is a exchange of gas. Right, what are we exchanging? We are exchanging the carbon dioxide in our body for oxygen in the lungs. Now, there are little structures on the lungs called alveoli. Okay, so these small little, I'll say grape-like objects that can contain the oxygen that we breathe in. So when we breathe in, there will be a high concentration of oxygen molecules in the alveoli, A-L-V-E-O-L-I, alveoli, and there's a low concentration of oxygen in the blood. Okay, and so there's going to be a net movement of oxygen molecules from the alveoli into the bloodstream. And the same happens for carbon dioxide, except the other way around, right? There's a high concentration in the blood, low concentration in the alveoli, so there'll be a movement of carbon dioxide. It will diffuse out. Okay, so let's talk about this in the roots also. Okay, so in the roots, in plant cells, because the cells in the roots are quite far away from, they're far away from the air, right? I mean, they're deep in the ground, let's say. There's no air in the underground. So the root cells also need to take in oxygen. So the oxygen will actually diffuse into the root hair cells, and then carbon dioxide will diffuse out of the root hair cells. So, so similar to in the lung. So this gas exchange of oxygen and carbon dioxide happens via diffusion. Okay, I'm so sorry that I missed this page, the previous lesson. Okay, so now that we've talked about diffusion distance, we can then continue with talking about osmosis. Okay, so why did I bring that up? Because the rate of osmosis is also affected by the three factors: water potential gradient, distance over which the water molecules need to move, and the surface area to volume ratio. Okay, for osmosis, it is not really affected by temperature, unlike diffusion. Diffusion will increase when the temperature increases. Okay, so how does osmosis affect living organisms? A cell is surrounded by a living, partially permeable cell membrane. Enclosed within the cell membrane are the nucleus, cytoplasm, and a mixture of various substances. Okay, so do you still remember the word protoplasm? Alright, these three things: the cell membrane, nucleus, cytoplasm, these are the protoplasm, yeah? Okay, so they just want to remind you that the cell wall of plants is fully permeable, and it will allow most substances to pass through. And this will actually change the behavior of a plant cell as compared to an animal cell. Okay, so let's first talk about what happens to a cell when we put it into a solution with higher water potential. Okay, let's talk about the plant cell first. When you put a cell into a solution with higher water potential, water molecules will move from a region of higher water potential, which is outside the cell, into a region of lower water potential, which is inside the cell. This will cause a plant cell to expand and swell. The plant cell is protected by the cell wall. So, the plants are supported by the pressure of water inside the cells pressing outwards on the cell wall. Okay, what that means is that when you have a plant cell, right, you put it into a solution of higher water potential. So the cell set has a certain water potential. Okay, let's say it is lower than the water outside of it. So by osmosis, water molecules will enter the cell through a partially permeable cell membrane, right? And this will cause the cell to swell up. The cell is now said to be turgid. The word turgid means that it is full of water and is harder than normal. The word turgid can only be used for plant cells. You cannot use the word turgid for animal cells. So as water molecules enter the cell, the vacuole increases in size and pushes the cytoplasm against the cell wall. The cell does not burst because it's protected by the inelastic cell wall. The turgidity of the cell with water is called turgor, or turgor, I think. The pressure exerted by the water in the vacuole is the turgor pressure. Okay, so this is what happens when you put a plant cell into a solution with high water potential. For example, distilled water or pure water. Now, if you compare this plant cell to the animal cell, we will see a huge difference. For an animal cell, it will swell up and may even burst when put into a solution of higher water potential than the animal cell, right? Because unlike the plants, it does not have a cell wall to protect it. So if you put a red blood cell into a solution with higher water potential, the cytoplasm of that cell has a lower water potential. So water molecules will enter through the cell membrane by osmosis. The cell will start to expand, and it will expand to the point where it will burst. The word burst, in medical terms, you call it as a cell being lyse, L-Y-S-E. Please write that here. L-Y-S-E, the cell has lyse or burst. Okay, so let's watch. Oh no, I'll say everything, then we watch the videos. Okay, so what happens to a cell in a solution of the same water potential? If it's put into the same water potential, the cell will not change in their size or their shape or anything, right? No change in volume, no change in mass, nothing, right? That's because the movement of water molecules is the same in both directions. There is no net movement of water molecules in or out of the cell. Okay, just a point there. They said no net movement, right? That doesn't mean that there's no movement at all. There's still some movement. It's just that the movement across, back and forth the cell, they cancel each other out. So there's no net movement. Okay. Now we talk about what happens to a cell in a solution with lower water potential. When a cell is placed into a solution with lower water potential, water molecules will move from a region of higher water potential inside the cell into a region of lower water potential outside the cell. And so the plant cell will decrease in size and become limp. Okay, the proper word actually is flaccid. Okay, I don't know why they wrote limp. Okay, so over here they wrote flaccid. Okay, so I'll use the word like flaccid. Okay, so inside the plant cell, we have cell sap, right? You know, got that good amino acids, sugars, salts, all inside the cell sap. Okay, that gives that cell sap a certain water potential. When you put it into a solution of lower water potential, what we see is that the cell sap will have a higher water potential than the solution outside the cell. So by osmosis, water molecules from the vacuole and the cytoplasm will leave the cell through the partially permeable cell membrane. Okay, so this, you can see the water molecules are leaving. And so the cell will decrease in size and become flaccid. Likewise, as the word turgor, you can only use the word turgid on plants, flaccid on plants also. Okay, you should not use the word flaccid on animal cells. Then, as the cell loses water, the vacuole will decrease in size, and the cytoplasm will shrink away from the cell wall. The shrinkage of the cytoplasm and cell membrane away from the cell wall is called plasmolysis. the cell is said to be plasmolysed. Important word also. A plasmolysed cell can be restored to its original state by placing it into water or into a solution with higher water potential. Okay, so the meaning of the word plasmolysed is simply pull away, that the cell membrane has pulled away from the cell wall. Okay, animal cell in a solution with lower water potential will cause it to lose water. The cell will shrink and spikes will appear on the cell. This process is called crenation. An animal cell will become dehydrated and eventually die when placed into a solution with low water potential. Okay, so animal cells not as hardy as the plant cells. Okay, so here we have animal cell, let's say red blood cell, and when you put it into a solution with low water potential, this will cause the water molecules to leave by osmosis through the cell membrane, and you will see these spikes. Okay, this is what I call as a crenated cell. Okay, let me just open the window, it's a bit dark. Okay, where was I? Okay, yeah, let's watch some videos. So here we have a red blood cell. Okay, this red blood cell right now is put into what you call as isotonic solution. Isotonic means equal in water potential. That's the meaning of isotonic. So one common example, our blood actually has a certain water potential, and our cells need to be at that particular water potential. Our blood is not the same as pure water. Right? It doesn't have the same water potential as pure water. So sometimes when you're feeling dehydrated, you are recommended to take what you call as isotonic drinks. Those isotonic drinks are specially designed to have the same water potential as your blood, so that it's easier for you to drink, and there are salts and everything inside that isotonic drink, so that your cells are going to be happy. Okay, so what happens to the cells in isotonic solution? I'm going to say in equal water potential solution. Okay, this is a what kind of cell? What do you think? It's a white blood cell. This is a red blood cell. Okay, changing the camera focus. Okay, so now they have changed the solution. Okay, you can see previously in isotonic, nothing happens, right? The cells are all fine. There's no change in volume, no change in mass, no change in length or anything like that. Okay, what happens? What is a hypertonic solution? A hypertonic solution is when there is let me just recall. Hypertonic solution is when there's too much salts inside the solution. Okay, so basically that solution is very, very low in water potential. Okay, so you can actually start to see some of the cells, right? They're actually starting to become crenated. You can see this one's actually become crenated already. This one also, right? Yes, starting to lose water. Right? Why is it losing water? Because the solution around it is very low in water potential, right? Let's say, for example, this is a salt solution, a very concentrated salt solution. Water molecules are leaving the cells and exiting the cell membrane towards the solution. Okay, so now they put the cells back into isotonic, equal water potential, so the cells are going back to normal. Okay, spelling mistake here. It's not distilled water, it's DI. So distilled water, properly, spell properly. Okay, distilled water, like I told you, distilled water is another way of saying pure water. Basically, all the salts in that water has been removed. So it represents the highest water potential possible. Okay, there's nothing in the world that has higher water potential than pure water. So when you put the cells into pure water or distilled water, you are basically putting the cells into a solution with very high water potential. So let's see what happens. Okay, what happened to the red blood cells? Okay, they didn't just disappear, they actually lyse, they have bursts, and so you can't see the cell membrane anymore. You can't see the cytoplasm anymore because it has already burst and the contents have spilled out into the solution around it. Okay, perfect. Okay, so now you can see the white blood cells have burst too. The white blood cells are a bit more obvious because they contain certain granules, certain enzymes inside of the white blood cell. And so when it dies, then all this stuff is all spilled out and you can actually still see it. Okay, so why did the white blood cell burst or lyse? Okay, let's use what I call as a osmosis statement. Okay, I'm gonna say this in a certain structure. Alright, I call it my CMOPS structure. C for compare, M for movement, O for osmosis, P for partial permembrane, and S for the size. So I start the sentence by comparing. There is a higher water potential inside the white blood cell as compared to the solution outside. Therefore, there is a net movement of water molecule. Did I just say it wrong? I just said it wrong. My goodness. I'm a bit tired now. Okay, so there is a, let's repeat from the start. There is a higher water potential in the solution as compared to the lower water potential in the cells. Therefore, there will be a net movement of water molecules into the white blood cells through the partially permeable membrane by the process of osmosis. Okay, so I've already said C, I've already said M, movement, I've already said O, osmosis. Okay, so what's the P? Right? Through a partially permeable membrane. Okay, the last S is size, or what happened to the thing. Therefore, the white blood cell will increase in size and lyse. Okay, so let's say that all together in one shot. There's a higher water potential in the solution as compared to the lower water potential in the white blood cell. Therefore, there is a net movement of water molecules through the partially permeable membrane and into the white blood cell by the process of osmosis. And this increases the size of the white blood cell. Eventually, it would lyse. Okay, of course you can rephrase your statements a bit, to make it more suitable to how you like to say it, but keep the general structure there. Okay, alright, now that we're done with blood cells, let's talk about plant cells. Okay, this time I'll let you just watch the video.
This is going to be my take on the red onion in fresh and salt water. So here I have a slide. I'm going to set that to the side, and I'm going to pick up a red onion. Now there are two layers that you can peel off of this. The first layer that I'm going to show you is the wrong layer. It's the inside one. It peels off super easy and it has no purple in it. You do not want that. The layer you want, and you're going to listen here, that sounds like tape getting peeled, and it should have purple on it. I'm going to place that on my slide, and there is an extra chunk there that's going to pose a problem. So I add some fresh water first, just a dab with my finger, and as I add my cover slip, it does not sit flat. It actually is raised off of there, and you do not want that. Smushing it is not going to help. So I need to cut off that extra little white piece that's there. There we got it. And now my slide is laying flat. So So now I'll place it under my microscope under low power,
Stage all the way up, power on, and center my slide. I'll turn on my microscope, and
there it is. Now as we look at this slide, we can see the cell wall, which is the dark outer layer.
The purple section is going to be our cytoplasm.
And inside there you can actually see little circles, and those circles are the nuclei that are inside that cell. So now I'm going to make up some salt water, and this is the technique we use to remove that water I added on earlier. You put the salt on the right side, and then I place a paper towel on the left side, and it's going to draw that salt water through. And as you see, this is sped up. It does dramatic changes to the cells, and you can see that that salt water draws the water out of these cells, and you can see them shrink down. The cell wall remains the same size, but the inside shrinks down, and we do call that plasmolysis. And that is a shriveled up onion cell in salt water. Now I'm going to remove the salt water with the same technique, but this time adding fresh water, and we should see these cells swell back up, back to their original size. And you can do this back and forth over and over, where you make them shrink and then fill them back up, and that is just osmosis working inside these cells. So I hope that made sense to you guys, and you enjoyed looking at that. Thank you.
Okay,
See that that's
I thought that was actually quite beautiful. So how would you write an osmosis statement for a plant cell that has been put into a solution with lower water potential? So for example, you put this plant cell into salt solution, very salty solution, then how would you write that osmosis statement? Okay, think about it, maybe say it out, try and verbalize it. If you're able to verbalize it well, alright, and can repeat it, consistently, then I think this is should be quite easy for you already. Okay, so let's talk about turgor. Okay, so we just saw the plant cell video. We saw that when you put water in the plant cells, they actually increase, they look like they're proper, right? And so that's called turgor, right? So basically that's when the large central permanent vacuole is filled with water. There's enough water inside that vacuole. Okay, so this turgor, it plays an important role in maintaining the shape of the tissues in a plant. The young stems and leaves of herbaceous and non-woody plants are able to remain firm and erect. Okay, so the word herbaceous means herby. Okay, so those kind of plants that don't turn into trees. Yeah, they just become herbs, like roses or that kind of stuff. Okay, so this is because of the turgor pressure within their cells. And when there is a high rate of water loss from the cells, they will lose their turgidity and the plants wilt. Okay, the word wilt means that like the plant that is becomes folded up. So the movement of certain plant parts are due to changes in turgor. For example, the changes in the turgor of the gut cells causes the opening and closing of the stomata. Okay, so what are stomata? Stomata are small little holes that are usually found at the bottom of the leaves. Okay, so imagine it's a leaf. At the bottom of the leaves, there will be many of these small little holes called stomata. This is where the carbon dioxide can enter into the plant leaf, and the oxygen, which is created through the process of photosynthesis, can actually leave the plant leaves. Okay, during the day, when there's sunlight and everything. Okay, so this is basically where gas exchange happens in the plants, as compared to humans, where that happens in the lungs. Okay, then what is plasmolysis? Plasmolysis is what happens when the cell becomes flaccid. The cells can be killed if they remain plasmolysed for too long. Thus, it is not advisable to add too much fertilizer around the roots of plants, because the soil solution will then become very concentrated and water molecules will move out the roots by osmosis. So here is a picture of a turgid plant cell. This is how plant cell should normally be like. Most of the time it should be turgid. When it's raining and everything, the plants are turgid. Then when it's very dry, it's very salty, the ground is very like salted around it, then the plant cells can become flaccid and plasmolized. Okay, I will choose to write these two words together. So when I'm writing my osmosis statements, I will always end it with these two words, right? The plant cell becomes flaccid and plasmolized. Okay, so example of turgor pressure, if you put water and everything for this plant, then the plant is able to stay up straight. There's turgor pressure. But when this plant is put into maybe salty sand or sand that is too much fertilizer, it will actually lose water and it will lose the turgor. It will cause the plant to wilt. Okay. We will do a potato strip osmosis experiment in the lab. So, okay, I will assign you a SLS to watch on how to cut potatoes. Okay, so yeah, okay, just quickly talk about this osmosis in a scape. Okay, what is a scape? It's basically a flower stalk called, example, a Chinese leek. So here you have a scape, right, the plant tissue, right, inside water and inside concentrated sucrose solution. Okay, what do you think will happen to them? So what's happening is that the outer layer is covered by a waterproof cuticle. So this green area is a waterproof cuticle and the white area is not waterproof. So basically this part will expand more than the green part. The white part will expand more than the green part. So what happens is that when you put this into the petri dish of water, there will be a net movement of water molecules into the plant cell, and this will actually cause that cell to become turgid. Turgid, right? It will expand in volume, in mass, and so the plant will be like formed like that, right? It becomes straighter, like that. It will actually like become straighter, bigger overall. So this area would expand and the whole area will like move down straight. Okay, whereas if you put it into concentrated sucrose solution, right, water molecules will leave the plant tissue and then the plant will actually shrivel up. So it will form like even more like a C shape. Okay, let's pause here and then we will continue the lesson and talk about active transport in the next tutorial. Okay, all the best.